Today, we're going to be talking about formal reports in Biology 107. At the end of Lab 3, you'll probably notice that you are being asked to write a formal lab report for this experiment, and it's worth 100 marks. This converts to approximately 12% of your final grade, so this is a significant part of your grade, and it's worth it to do well on this particular assignment. For the formal report, we're looking at several sections: a title, introduction, methods, results, discussion, literature cited, and appendix. And I want to talk a little bit about each of these sections. I will say before I get into this that there is detailed information on formal reports in Appendix A of your lab manual. I encourage you to look at Appendix A for that detailed information on what is expected of you. Here, I'm just giving you a little bit of an overview, but you do want to look at Appendix A for that detailed information. Overall, your lab report is going to be about 10 to 12 pages. That includes 5 to 6 pages of actual written material, double-spaced, and maybe 4, 5, 6 pages of graphs, appendices, calculations, those kind of things. So overall, 10 to 12 pages. This is a big assignment, and it's best that you do not do this assignment all in one night. You're probably going to want to work on at least one section a night for over maybe 4 or 5 nights, and then another evening, you're going to put it all together. Hopefully, you have time to ask me any questions if you don't fully understand what is expected of you. So let's take a look at what you need to do for each of these sections. I'd like to start off with the results section because this is the place that you should start as well. And the reason for that is the results are the basis of that experiment. So when you put together the results, you are making the graphs and you're taking a look at the experiment. It gives you a chance to think about what the trends are, what the conclusions might be, as you're writing the rest of the report. So hopefully that gives you the idea of the basis of the report. The results section is going to have 2 parts. The first part is a written part, and this is a paragraph form summary of the results. I'll talk about that in a minute. But this is basically a description of the trends that you observe in your graphs. And so the graphs, of course, are part 2. If you recall the experiment, we are making 3 graphs in this experiment. The first one is the standard curve, the second one is the temperature experiment, and the last one is the solvent experiment. So what the results section does not include is your rough data. The tables that you generated, these are going to go in the appendix as the rough data. I only need to see the graphs here. And any kind of discussion or interpretation of the results, we're going to save that for the discussion section of your report. So let's talk about this written section first, and then I'll talk about the graphing in a minute as well. So the written section, like I said, is looking at the trends of the results. So you have 3 graphs, so there should be, some people might say 3 paragraphs, 3 long sentences, but basically I need you to address each of those. You need to be brief, but also informative and specific. I'll give you some examples here. So take a look at this. Which is better, the first one or the second one? I'll give you a moment to read these. So hopefully you've concluded that the first one is better than the second one. Notice the second one is a good start. It's giving us good information about the high temperatures and the low temperatures of the graphs, of what you're seeing in the temperature graph. The first one, though, on the other hand, is much, much better for quite a number of reasons. First of all, it's referring to the actual graphs. You can see in brackets it has figure 2. Secondly, it's telling me not just the high temperatures, but what high temperatures. So 85 degrees or 65 degrees or whatever temperature we used. It's also telling me how much leakage we had at those temperatures. So you don't need to list and give what happened at every temperature, but the trends and a little bit of the data to back up about what you're talking about. So in this case, I'm being very specific about the minus 20 and the 85 degrees Celsius. So your written part is going to give the overview of the graphs, and then the graph part is going to be the actual graphs. So here's an example of a graph that you might see in one of these lab reports. So this is the temperature graph as well. So you can see I've plotted the amount of beta sign and leakage on the y-axis and temperature on the x-axis. And so I want to point out a few features in this graph. First of all, when you make the graph, you should have clearly defined data points. You can use diamonds, you can use squares, rectangles, circles, whatever. But just make sure they're very clearly defined so I can see where those data points actually are. Number two, you're going to label your axes, and the axes are going to include the units. Make sure you have the proper units. Degrees Celsius, not degrees Kelvin, for example, and so on. Number three, you're going to have a title, and the title is going to start off with the word figure and then be followed by a number. So this could be figure 2. This is lab 3, so I'm calling it figure 3-2. Either of those work. If you want to use A, B, C, that is fine as well. The figure title is going to be below the graph. This is the standard in biology. If it is a table, the figure title goes above. And make sure the title is describing what is going on in the experiment. So we're looking at beta cyanide leakage when we're exposing the beetroots to different temperatures. Make sure you don't use the word beetroot, but you get the binomial name of the organism, which in this case is Beta vulgaris. So the graph, you can make it in Excel or another software, or you can make it on graph paper, but make sure you follow these tips around the graphing. Next, we want to take a look at the title of the experiment. This is worth 2 marks. Make sure that your title has the binomial name. So there's the binomial name of the beetroot, Beta vulgaris. Make sure that's in italics because that's the proper way to do scientific names for organisms. Make sure the title is informative. So you're talking about what was done. So the word temperature, the word solvent had better be in that title somewhere, and the species name, and what were we looking at? We were investigating the damage to membranes, something along that lines. So make sure that you have a descriptive title. Here's a couple of examples of some titles on a different experiment. So you can see the first one, effective cold temperatures on humans, not bad. A little vague. The second one is much better, effect of cold temperatures and exams on the mental health of young adult Homo sapiens. So notice it has the species name, binomial name, Homo sapiens. It's giving me much better specific information. What kind of effects on mental health. And so I feel like that's a lot better. Next, let's talk about the introduction. So your introduction should include 3 things. And so this could be 3 paragraphs. Usually this is about a page and a half on these kind of reports. So some background information, so maybe a little bit on what a membrane is and what it does. So that would be a sentence or 2, and why we might be investigating membranes in cells. The objectives kind of lead into that. The objectives of the experiment specifically, we're looking at temperature and solvent effects, and then your hypotheses. So you should have 2 hypotheses, one relating to temperatures and one relating to the different solvents that we used. Let's talk about the methods. So the methods section should be very short, and you're doing 2 things. First of all, you're citing the lab manual. So you're telling me where I can find the detailed information. So this is how I want you to do the citation in this course. There's no author for this lab manual, so we're going to say that Keano College is the author. And so you put this in brackets, comma, followed by the year, and then you finish the sentence. We've talked about citations already. I'll talk about them again in a few minutes in this video, but make sure that your first sentence is citing the lab manual. Secondly, a concise description of what was done. I would like you to fit this in 1/2 page double spaced. So make sure you make it clear what was done in the experiment, but you're not going into a crazy amount of details. Just give me the essential details. Let's take a look at an example here. So which is better? Take a look at A, B. I'll give you a moment to read that. I'm hoping that you've concluded that the first one is much better. So first of all, let's take a look at the second one. It's not bad. It's giving us some good information, but it really is a lot of information that is not necessary for me to repeat the experiment. I don't need to know that a razor blade was used to cut the beets. There's many ways to cut beets. I don't need to know about all of that stuff. If I want to know about that stuff, I can really look it up in the lab manual. You can see the first one, the first example, it does cite the lab manual. And like I said, that should be your first sentence. You can copy that one word for word if you like. And then it goes right into what is going on in the experiment. So you can see it's talking about the beetroot plugs, the size of them. It's telling me the specific temperatures and how long it's exposed to those temperatures. And so hopefully you might be able to do each procedure in one sentence. That is my goal for you, that you can do this in 1/2 page. It can be specific, but concise at the same time. Okay, let's talk about the discussion section. So this is where this is worth, I think, 1/3 of all of the marks of this report. So this is where you want to discuss, interpret, elaborate, and you want to be thorough. This is where you are trying to convince me that you understand what is going on. I've guided you through how to answer the discussion by giving questions in the lab manual that you need to answer. And so that's what you need to do. Go through those questions, be thorough, relate them to things we've talked about in the class, relate concepts to, say, the fluid mosaic model. You can look up the textbook, and that's what I want you to do in this section. So this is usually at least 2 pages of written material. And like I said, it's worth a big part of your grade, so you want to be thorough and make sure that you are answering the questions completely. Here's another example of a couple of sentences, and you can see that the second one is better than the first one. Again, giving that specific information, referring back to your results, all those are good habits to get into when you're writing technically and scientifically. So notice that the second one, I can go on and on, right? I'm trying to explain things, what I think has happened, and don't just come up with answers out of your head. You can Google this or look at your textbook and get information about why what is happening is happening. And if you want to be really good, you're going to reference this as well. This is not expected necessarily in Biology 107 that you're going to be citing and referencing every single sentence. I'll talk about citations in a minute, but this is a way to give support to your conclusions where you are showing that somebody else has come up with the same conclusions. So one thing I just want to point out is sometimes there is confusion regarding the solvents. Here are the 4 solvents we use: water, methanol, ethanol, propanol. If you take a look, there is a clear trend with the molecules used in this particular, for these solvents. If you take a look, we're getting more and more carbons as we move to the right. Compare this to another molecule, fatty acid. So fatty acid has a whole bunch of carbons and hydrogens attached to it. So one thing you probably know about that fatty acid is that it is hydrophobic and nonpolar. And that's the trend that we're looking at with these solvents. As we go from water to propanol, we're going from most polar to least polar. Okay, so the reason why I'm pointing this out is sometimes people get confused and think that sometimes these solvents are acidic or something like that. They are not. They are polar and non-polar solvents. So lastly, I want to talk a little bit about citations and how we're doing that. And we have talked about this already before and when we're looking at the Lab 3 pre-lab. So citations, you should have 2 in your report. You can have more, but I'm looking for 2. First of all, you need to have a scientific paper cited in your report. You can do this in the introduction or the discussion. Number 2, your lab manual should be cited in the methods section. So remember that citations are done like this. They're at the end of the sentence. If you have one author, it's the last name, comma, followed by the year. If you have 2, you can use both last names. If you have 3, use et al. Here's an example within a sentence. And notice in this case, I actually put the citation earlier on because I referred to the author. So you can do either way. The lab manual has no author, and so we're going to use the college name for the author, and the citation is going to look something like that. Notice the period is after the brackets because the citation goes with the sentence that you used it in. The works cited section, this is where you're going to get the complete bibliographical information of your references. And so the standard format for a journal looks something like this. You've got the last names, followed by the year, followed by the title in sentence form. The journal name, the volume is bolded, then the pages, and the digital object identifier. So here's an example from the literature. You can see there's all the names. Notice all that punctuation and all the correct information. Here's the lab manual. You can get that right out of your lab manual in Appendix A. Have that listed there. Don't forget the proper year, 2023. And just a reminder that every punctuation detail is important. So a couple other general things. First of all, be specific. Always give the amount of detail so that I fully understand what you're talking about. If you're going to be vague, it's really hard to understand what you're talking about, particularly if I have never done this experiment before. I want you to be specific. That's important for technical scientific writing. Number two, try to make your report clean and professional. One way to do this is to use special features, right? Special characters. So for example, for carbon dioxide, you want to use the subscript feature. For micromole, you want to use the special Greek symbol. For degrees Celsius, you want to have the proper degrees symbol instead of a 0 or something like that. So learn how to use these features in Word. If you don't know how to do them in Word, drop by my office anytime and I'll show you how to do it. So I'm hoping that you found all of this useful for the Lab 3 formal report. You can, of course, drop by my office anytime. If you have a rough draft that you would like me to look at, I can give you a little bit of feedback on that. Make sure you use this grading scheme as a little bit of a sort of a checklist that you're going through and making sure that you include everything.